Good evening, everyone. My name is Mahmoud Summers, and I'm the host for today's lecture. We thank you for joining us for the second lecture of the Oxford SARS-CoV-2 lecture series. This is a weekly online webinar series that will host a wide variety of keynote speakers from around the world that are at the forefront of COVID-19 research. Our objective for the webinar is to invite brilliant minds to give talks that are research-focused and on a wide range of subjects to foster learning, inspiration, and innovation, and provoke conversations that will dictate the future of healthcare. Before we transition to the lecture, I'd like to briefly introduce the series, upcoming webinars, and our guest speaker for today, Professor Adrian Hill. In addition, I would like to highlight the procedure in which the viewers may submit questions for the guest speaker, which will be addressed towards the end of the event during a short Q&A session. This event series is hosted and organized by the Oxford University Personalized Medicine Society, a student and faculty-led organization that works to weigh the benefits and detriments of precision healthcare in modern-day medicine. We would like to take this moment to thank our sponsor, the Oxford Center for Personalized Medicine, which is a partnership program between the Wellcome Center for Human Genetics and St. Anne's College, University of Oxford. Throughout the year, we collaborate to hold guest speaker events encompassing a diverse array of subject areas, interdisciplinary symposiums, and field trips for all affiliates of the university. If you are a current member of the university and would like to get involved, please register using the above displayed QR code. I would like to highlight that membership is free, unlike many other societies at the university. If you have any questions regarding the society, please email us at oxmed@ox.ac.uk. For today's lecture, Professor Adrian Hill will give a talk entitled A Rapid Vaccine Response to COVID-19, Progress and Prospects. Professor Adrian Hill is the director of the Jenner Institute, co-director of the Oxford Martin Program on Vaccines, and professor of human genetics at the University of Oxford. He's also the head of a research cohort at the Wellcome Trust Center for Human Genetics, which studies genetic susceptibility factors for common bacterial diseases. His work is mainly focused on designing and developing vaccines for infectious diseases prevalent in developing countries such as HIV, AIDS, malaria, and tuberculosis. Previously, his research group conducted the first clinical trial of an Ebola vaccine, and they developed one of the most promising potential vaccines for malaria, which is currently in large-scale clinical trials in sub-Saharan Africa. Recently, his laboratory group has pulled ahead in the worldwide race for a vaccine to stop the coronavirus. At this stage, they have completed the initial human clinical trials, and the results appear to be promising. If the rest of the studies go well, they hope to have the first doses of the vaccine ready by September. For upcoming webinars pertaining to the lecture series, we have next week, Wednesday, May 27th at 5 p.m. British Summer Time, Professor Dan Baruch of Harvard Medical School and a director for the Center for Virology and Vaccine Research at the Beth Israel Deaconess Medical Center will be presenting a lecture entitled Natural and Vaccine-Induced Immunity to COVID-19 in Rhesus macaques. The following week, on Wednesday, June 3rd at 5 p.m. British Summer Time, Dr. Seema Yasmin of Stanford University and a director of the Stanford Health Communications Initiative will give a talk entitled Misinfodemics, Pandemics of Microbes, and Misinformation. To register for these events, please RSVP using the Eventbrite to ensure your spot for these lectures. For later dates, we have Thursday, June 4th at 5 p.m. British Summer Time, Professor Karai Nagyao of Stanford and the Director of Clinical Research for the Division of Hospital Medicine will be presenting a lecture entitled COVID-19 Immunity in Progress. In this lecture, she'll be discussing some of her experiences in successfully running an NIH-NIA ID-funded remdesivir clinical trial. Then on Friday, June 19th at 5 p.m. British Summer Time, Professor Scott Boyd of Stanford and the lead researcher behind a novel antibody test will be presenting a talk, the title of which will be announced at a later date. Again, to sign up for these events, please RSVP using the Eventbrite to ensure your spot for these lectures. To submit questions for the live Q&A at the conclusion of the event, please use this link/QR code to navigate to a Google form that will allow for you to submit questions. If you'd like to submit multiple questions, please use the form to make multiple submissions. Thank you. And now, without further ado, we would like to welcome Professor Adrian Hill to present his talk. Please stand by as we transition to the guest speaker. Thank you so much.
Thank you very much, Mahmoud. It's a delight and an honor to present to this very large number of people. I will be talking about a candidate vaccine for COVID-19 and telling you a little bit about how we came to make this in January of this year and progress it rapidly into clinical trials and, as will be announced tomorrow, to an even larger possibly final clinical trial for this vaccine. I should emphasize that this is the work of literally hundreds of people that I'll be summarizing quickly. There are 5 principal investigators who have come together at the Jenner Institute to put this rapid response program together. The project lead is Professor Sarah Gilbert, who designed and initiated the work on the vaccine. Professor Pollard, who runs the current major clinical trial. Sandy Douglas, heavily involved in scaling up manufacture, which we think will be critical. Teresa Lamb doing preclinical work, and I get the job of telling you about it. So without further ado,
could you please share screens?
Can you not see it? Okay.
No, I can't see it. Yeah, thank you so much.
Try that once more. Okay.
Thank you.
Sure.
There we go. Thank you.
Tells me we're sharing. Good. So those are the 5 PIs who I've just mentioned. And of course, our institute, the Jenner Institute, goes back to Edward Jenner, the inventor of vaccination over 220 years ago. And we like to think that we're still doing some things in engineering fashion. We have a particular focus on global health, on translational research, moving things rapidly into the clinic. So we were set up for this rapid response. And we also partner on veterinary vaccines in a One Health strategy. We are based about a mile and a half outside the centre of Oxford at the Old Road campus, where we have 3 key facilities, the Jenner Labs on the Old Road campus, and then on the Churchill Hospital site itself, we have a clinical centre, the Centre for Clinical Vaccinology and Tropical Medicine. And then unusually for a university, right outside the door of that, our clinical biomanufacturing facility, where we can manufacture vectored vaccines for clinical trials. And we've been doing that since 2005, and that proved to be very important in the COVID response. This is what we generally do, or what we're there for, making new vaccines against difficult pathogens. For example, malaria has been the largest effort in the Institute, tuberculosis, hepatitis C. I won't read through the whole list, a universal flu. Ebola is on there. We were heavily involved in Ebola back in 2014 and 2015. And you can see down at the bottom, there's now an increasing interest in outbreak pathogen vaccines that I will be talking about. The other thing you'll see is that we've got into a lot of clinical trials, over 50, phase 1 and phase 2. Phase 2B is a field trial. We've never yet got to phase 3, but that may be about to change. This is Sarah Gilbert's program, a very ambitious one set up in 2014. You can see that Ebola is on there, but also nearly all the other pathogens in the World Health Organization's list of outbreak pathogens that we would really like to have good vaccines against. And as you can see on the slide, some of these have gone further than others. Many of them are already in the clinic, like chikungunya and Zika, and several others coming along. But COVID, of course, is not on there because these were the vaccines we were doing back in 2019, which seems a long time ago now, and everything changed in January of 2020. So I'm going to tell you a bit about the candidate vaccine that this talk is about and the technology underlying it. So that cartoon on the left is of an adenovirus. You've probably all had adenoviral infections. They cause common colds in humans. Then you look a bit more carefully, they're about 90 nanometers in diameter. The nucleic acid is double-stranded DNA, and these, of course, replicate in the nose. If you do some genetic engineering and take out the genes in the so-called E1 region, possibly E3 region of the genome, the whole genome is about 35 kilobases, it's 90 nanometers in diameter, that particle. you find that the virus can no longer replicate. And that's crucial for the safety of this vaccine. And into the place in the genome where we took out those critical replication genes, we insert whatever antigen we want to vaccinate against. And this might be an antigen from the malaria parasite, from the TB bacterium, or in this case, from the COVID-19 SARS coronavirus 2. And what we do is we use a very strong promoter from CMV to drive that transgene, as it's called, so you get very high level expression of the virus. Now you inject the virus, or the vaccine virus, intramuscularly in pretty large amounts, maybe 10 to the 10 virus particles. A lot of it goes into the cells where it turns on the transgene and starts expressing the, in this case, COVID-19 spike protein, because that's the antigen from COVID-19 that we and nearly everybody else designing 100 or so coronavirus vaccines, I'm told at the moment, has chosen. The one twist we put on this back in 2007 was, instead of using a human adenovirus, we accessed one from a chimpanzee, so it's a simian adenovirus. And the reason for doing that is that we all have antibodies and some immunity against several human adenoviruses. And if you have that and you're vaccinated with that particular adenovirus carrier, you find that the vaccine works less well. It can be cleared by the immune system, but happily we don't get simian adenoviruses where nobody's infected by them. There's a minimal anti-vector immunity in the human population. So these are more promising, more successful viral vectors. As I've said there, we put the antigen under a strong promoter. It's not expressed in the virus itself. It's only when it gets into the arm that you express the antigen. And very importantly, that high level of expression intracellularly gives you not just antibodies, and as I'll show you, neutralizing antibodies, but also very good T-cell responses. And in fact, this technology was developed in the noughties decade, if you like, for primarily vaccines targeting cellular immunity, and it's often used today for therapeutic vaccines in, for example, cancer. It's relatively easy to manufacture on a standard human embryo kidney 293 cell line, commonly used for making other biologicals. And crucially, for the rapid progress in the COVID trial program, this exact vector had been used in 12 previous vaccine trials, phase 1 studies, in over 300 people, and had been safe with a very consistent safety profile and very strong immunogenicity after a single dose. So that encouraged us to believe last January that this would be a useful vaccine platform, if you like, for taking forward with the COVID virus, as well as with many other viruses that we've used it for, like Zika and chikungunya in recent years. So this slide just gives you a sense of how new or not this chimp vector or CHAD technology is. It goes back to 2002, first used at the Wistar Institute by a gene therapy group, and shown to be a good vaccine for rabies in mice. Then it was taken up by an industrial group in Italy, then part of Merck. We collaborated with them from 2005 onwards, and in 2007 managed to get the first CHAD vector into a clinical trial, actually for malaria, and that looked pretty safe and immunogenic in the very first trial. We worked with them for several years. We made our own viral vectors. The OX means they were made in Oxford, so you have the ChAdOx1 and ChAdOx2. This is the first one we made. The Italian group were bought out by GSK. They're now part of that pharma company. And we worked with them during the 2014-2015 outbreak to take a chimpanzee adenovirus all the way to West Africa to large-scale trials in several countries where it was very immunogenic, but before we had time to test it for efficacy, the Ebola outbreak ended, which was disappointing for the clinical trial team, very good for West Africa, but it taught us a lesson that unless you move very quickly in these outbreaks, you may be left with no data on efficacy. In 2016, Sarah Gilbert and I co-founded a company called Vaccitech here in Oxford, to exploit the Chimpanzee Adenoviral platform. They have been involved in the early stages of making the vector that I will talk about and progressing it. But since 2007, there have been a lot of clinical trials now in malaria, 5 or 6, hepatitis C, TB, flu. I won't read the whole list, except point out that Zika, chikungunya, and MERS are on that list. And MERS is important in this story because it's the other circulating coronavirus that causes disease in humans, Middle East respiratory syndrome, so it's in Saudi Arabia and some neighboring countries. And Sarah had already taken that vaccine into the clinic through a phase 1 trial in Oxford, shown it was safe and immunogenic, and that is currently in a clinical trial in Riyadh in Saudi Arabia. So in total, the experience with this technology is of over 6,000 humans dosed with 22 different vaccine candidates, none of them licensed yet, but some progressing very well, but crucially, a very good safety profile. And because the vaccine platform had been in so many trials, many of them in the UK regulator was very open to hearing more about another chimp adenovirus vaccine that we want to progress very quickly, because the regulators as well as the investigators were familiar with this vaccine technology. So what's good about it? Why are we using a ChAd vector? Well, firstly, we expect it to be a single dose vaccine. We're not 100% sure of that. It might need a second dose, but probably a single dose will do. I'll show you some evidence of that in the animal models. And of course, in an outbreak situation, a second dose is not ideal. You have to manufacture twice as much of the vaccine, and it takes longer to generate immunity. Safety record I've mentioned, we can manufacture it fairly quickly. It induces both cellular immunity, which we think is probably relevant to protection in COVID, as well as good antibodies with a single dose. You can manufacture it, and it has been manufactured at significant scale, not millions or hundreds of millions of doses, as people are trying to do now, but certainly at larger scale, particularly by a company called, well, GSK initially, and now Janssen are doing that for their own adenovirus vectors. There's very little anti-vector immunity to the vaccine itself in humans because it's a chimpanzee virus, so you can reuse it and probably reboost. And if you need to use the vector for another disease, there are quite a few different chimpanzee vectors around and in the clinic from companies small and large, including GSK, Merck, and Pfizer. There are also, I should say, human adenoviral vectors being developed very notably by a Chinese company, CanSino, I'll come to those, developing an adenovirus 5, which is a human adenovirus. And then, of course, Janssen, part of Johnson & Johnson, developing Ad26, which you'll hear a lot about from Dan Baruch if you tune in next week at the same time. This is just an indication of what this type of vaccine does with a single dose in a mouse. On the left-hand side of the screen, as we look at it, if you put an irrelevant green fluorescent protein antigen into the vector, you get no T-cell response. If you use the SARS-CoV-2 spike antigen, you get a very strong T-cell response measured in an ELISpot assay, and the numbers here are spot-forming cells per million splenocytes in the mouse spleen. And you can see that in different strains of mice, VALV-C or in CD1 outbreak mice, you still get well over 1,000 antigen-specific T-cells per million splenocytes. Over on the right-hand side, you can see the same sort of data, but this time for antibodies in mice. So the irrelevant vaccine gives none, and the SARS-CoV-2 gives you good titers of antibodies, both in the VALV-C mice and in the CD1 mice. So that's a good start, but it's not hugely difficult to get good immunity in mice, much harder in humans, and somewhere in between is getting good immunogenicity and efficacy in non-human primates. So let's move on the right-hand side of this slide to the NHPs, the non-human primates or macaques. And here you can see that in this first experiment, and we only had time to do one because there was a race to get into the clinic, at 14 days after vaccination, you're getting pretty good antibody titers after a single dose in the macaques. And at 28 days, the day they were challenged on, those titers are slightly higher, if anything. There were also neutralizing antibodies, which we'll come to in a moment. And the reason for doing this study in non-human primates that was done in Montana in the Rocky Mountain labs by the NIH group was, yes, we were interested in seeing some efficacy, but the real reason for doing it was we wanted to reassure ourselves and regulators and everybody else that we weren't going to see any adverse reactions after the vaccination, because with the SARS-1 virus outbreak in 2003, people made vaccines that could actually make the disease worse. Now, they were very different types of vaccine with alum as an adjuvant, but nonetheless, that's been a concern of regulators. So they asked if we could do this. They didn't require it, but they were much happier that we did. And lo and behold, when we challenged these monkeys, there was no sign of immunopathology. So let's have a quick look at that experiment, which is summarized on this slide here. We had 6 macaques immunized with a single dose of the vaccine, half the human dose, and 3 control monkeys. And they had a very high dose challenge. Now, this is one of the issues, because this had been done once before in history, meaning a month earlier, anywhere, a dose had to be picked. And it was a high dose deliberately, so that if there were to be immunopathology, we would pick it up. Remember, this was a safety study primarily. What we saw was on clinical signs, looking at the vaccinees in red and the controls in blue this time. Yes, less clinical signs over numerous days in the vaccinees than in the controls. Very strikingly in the lung, this is the bronchiolar alveolar lavage fluid. Looking in the controls in blue, 6 to 7 logs of virus. This was a heavy challenge on day 3, but down in the vaccinees here, apart from one animal, almost nothing. Day 5, very similar. You're getting a 3 to 4 log reduction in viral load, whether you're measuring genomic DNA or more replication competent subgenomic RNA. But interestingly, and this was commented on by one reviewer, in the nose swabs, we didn't see very much, and we don't know why that is. Maybe it doesn't protect in the nose, or much more likely, we just swamp the nose with the amount of virus that we put in there. If you look in detail in the lungs in different places, you can see some striking efficacy comparing the blue to the red, particularly here with the subgenomic RNA summarized there. So these animals, essentially all the vaccinees were well, the controls were not. We're doing other non-human primate studies, some in England at Public Health England in Porton. We're doing ferret studies that are ongoing, and we're getting preliminary data on those, both in the UK and in Australia. But the key immunological finding so far for the immunologists amongst you is that, as always with a chimpanzee adenoviral vector, you're seeing a Th1 and not a Th2 type immune response. And that's important from a safety point of view, because the alum adjuvant induces a Th2 type response, and that was associated with immunopathology. So we did have the data from the non-human primate study before we started the trial on the 23rd of April last month, and the Data Safety Monitoring Board were very reassured by that. So what does the first and indeed the second clinical trial look like? Well, we started last month, almost exactly 4 weeks ago, in a total of around about 1,100 subjects. And that's a very large number, a very unusual phase 1 trial. In fact, it's a phase 1/2b trial, because there's an efficacy component in there, as well as the usual safety and immunogenicity evaluations. This was in, or is in, healthy adults, 18 to 55 years of age. We didn't go above that age because this was the first time in humans. We have measured a lot of things, obviously safety, but also ELISA antibodies, ELISpot T cells, neutralizing antibodies, and these individuals are being followed for efficacy. Now, it's quite a small trial to follow efficacy for COVID, just 1,100 subjects, but they will add to the phase 3 trial that will be starting soon to give much larger numbers. We're also looking at a prime boost protocol, 2 doses of the vaccine 4 weeks apart, just to check that that isn't hugely different from giving a single dose. If it is, we'll consider swapping to a 2-dose regime. But what we're going on to do very soon, and we're just starting recruiting around about now, is a phase 2/3 trial, phase 2 being older adults, shown here as 56 to 70-year-olds and over 70-year-olds. Later on, a very small number of children, not really for efficacy, just for safety and immunogenicity. But the main study, as you can see on the bottom line here, is in over 18-year-old adults, right up to over 70 years of age. There will be 10,000 subjects in that trial, 5,000 receiving the vaccine and 5,000 receiving, not exactly a placebo, another irrelevant vaccine that has a similar reactogenicity profile. So this is a blinded, randomized control trial done across 2 trials in a total of over 11,000 subjects. The primary efficacy endpoint will be symptomatic disease along with PCR positivity, either in the nose or the saliva for the SARS-CoV-2. We will have a secondary endpoint where we swab people regularly every week to try and increase the power of the trial. We might have more endpoints, even if people are asymptomatic. And then a further endpoint, now possible with serology, is to pick up the cases who had asymptomatic disease because they seroconvert, and that might give us a few more cases. Remember, most people are symptomatic, but there are a small number who are asymptomatic. And the reason we're so focused on numbers is, of course, because happily, the pandemic is declining, at least in the UK. We will have 18 centres, as it shows there on the slide, from Scotland to Wales to many in England. And the reason for that is to try and maximise the number of incident cases, because we don't know in 3 weeks from now where most COVID is going to be in Britain. It might have moved back to London. It's certainly not in London at the moment. Might have moved up to Scotland. We want to spread our bets, if you like, to try and get as many cases as possible. And of course, it's crucial to get enough cases. If we only get 5 in the whole study, we have no power to see efficacy. If we get, say, 25, we're probably there to get a useful readout. And the study will end when there is, of course, a significant difference in case numbers between the controls and the vaccinees. So nobody knows when that will be. These individuals are being followed, blinded, except for a data safety monitoring committee who would bring the study to an end when there was a clear, positive, protective result found. We're guessing that might be around about August time. It might be before, if the cases don't decline as quickly as we expect, or might be later if we run out of cases. There are those safety groups in there as well, as it says on the slide, really for immunogenicity. We may not have as many cases in older adults, say over 70s, to declare success in that age group. But if the immune responses are the same, that would be very reassuring. So that's the design that has gone through, obviously, an ethical committee and a lot of iterative discussion with the regulator in the UK, the MHRA, who have been absolutely fantastic, advising us as well as regulating us, and have approved this trial to go forward in the very near future. But of course, there may not be enough cases. So what do we do if we don't get a result in the UK? Well, there are a lot of backup plans being discussed, all of which essentially are doing trials in other countries where there may or may not be more COVID in a month or two's time. As some of you are aware, there's a lot of transmission in Brazil, and unfortunately the incidence seems to be still increasing, so that may be a good place for a trial. None of these are confirmed. These are places we're in discussions with, where investigators have approached us saying they would like to do a trial. In India, a lot of disease as well. South Africa, Johannesburg. The university has a unit in Kilifi on the coast of Kenya. They're gearing up as well and seeking approvals. And of course, in the US as well, where there is a lot of variability in incidence, some very high incidence states, New York and Massachusetts and so on, some with very little. And it may be that by the time we end up vaccinating in the US, assuming that we do, the peak of the incidence will be somewhere else, particularly as lockdown is being lifted in several states. So that's not just a backup program for getting efficacy, that's a way of getting more safety data in more than the 5,000 or 6,000 people in the UK, and we believe that will help with registration in various countries. So let me just switch gears a little bit now and ask, what are we really trying to do? Because back in January, this was a little outbreak in Wuhan that a few people had heard of outside of China. And Sarah Gilbert was one of those because she runs the outbreak pathogen program and picked this SARS-CoV-2 as a virus where we could illustrate that we could make a vaccine very quickly. She had no idea this was going to go global. But by the time the vaccine was made and in mice, it started looking very serious. Today, people are talking about trying to vaccinate massive numbers of people. There are 7.8 billion of us on the planet. So even if you omitted all the children and the younger adults and teenagers and said they're going to be okay, they don't need a vaccine, and not everyone would agree with that, you still need about 4 billion doses. If you excluded a small number of regions where there's very low incidence and may not be much, you're still up at 3 to 4 billion doses. And you'd like that this year rather than next year. Now, not only has nobody made a vaccine at that scale in a year, no vaccine has ever been supplied at that scale in any year, even if you add up the combined output of different companies. So a typical amount of flu vaccine, for example, would be made by one company, would be a few 100 million doses in a season. And that's a race because they get 6 months to manufacture it. Could you possibly do that for COVID? Because some people want this to happen and are willing to fund it. Well, let me pause that question and tell you what we did in our own small way in manufacturing and how we got to where we are. To manufacture any adenovirus vector vaccine, you will use HEC293 cells or some variant of that. You need to make, as always, for good manufacturing practice, seed stocks, which takes you a while. You have to qualify them and characterize them in various ways. Certainly can't jump straight into vaccinating people. We sequence the whole virus. It's only 35 kilobases. And then crucially, we had our own facility outside the door run by Kath Green, the clinical biomanufacturing facility, who downed tools on what they were supposed to be doing and started going with this. And within about 6 weeks, they had the first fill. In the middle of April, you need to do assays after that, which is why it was late April by the time that we were able to get approval to vaccinate anybody and do all the risk assessments and everything else that you need for a new vaccine. In parallel, one of our contract manufacturers in Rome called Advent started on a larger batch. They produced 6,000 doses, which were ready just a couple of weeks ago, so they're ready for use now. And in parallel, we started looking at larger manufacturers, booking in slots at risk, paying for them, reserving them. 3 of them are Cobra and Helix. Cobra up in Kiel in England, Helix in the Netherlands, and OXB is Oxford Biomedica in Oxford. And they started scaling up from 50 liters to 200 liters. We want to go further. And the great news really has been the yield so far looks excellent. If you get 8 doses of vaccine per mil of bioreactor fluid, and you can go up to a 10,000 liter bioreactor, you can see that then you're really motoring and getting tens of millions of doses. We're going to go to 2,000 liters fairly soon. We haven't got there yet. You can't do this thing in a couple of weeks. And we're partnering with lots of large-scale manufacturers for global supply. I'll come back to this. But the point I really want to make here is that it's said there are about 150 COVID vaccines in development. Very few of those have got to the clinic. A few more are definitely going to get to the clinic. These are the leading ones that I'm aware of. And lots of the journalists out there like to characterize this as a race between lots of different rival universities, companies, institutes, whatever. It is a race. It's not against each other. It's against the virus. It's against the outbreak. If we run out of cases, we'll have no efficacy. And I see the others ideally as allies rather than competitors, because thinking of that 3 or 4 billion doses and where they're going to come from, several of these vaccines are going to have to work if we're going to get anywhere close to that, because a billion doses over the next 6 to 9 months would be absolutely remarkable for any one manufacturer. But the good news in this perspective is that you can classify these different vaccines that are competing in a sense into groups according to the technology base, and there's ours. If ours works, I think there's a really good chance, for example, that Janssen's vaccine will work. It's an Ad26, it's an adenovirus, same sort of technology, same sort of manufacturing scale possible. They're a bit behind us in terms of months there. CanSino were some weeks ahead of us. They've got an Ad5, but these produce broadly the same sort of immune response. They've got the same antigen, all of them, very tiny tweaks of differences. But I think if one works, the others will be pretty similar. That's good. Janssen are a large-scale manufacturer. If ours works, they can do some of the manufacturing and sell theirs as well, would be my personal view. On the other hand, if you're a fan of new RNA vaccines, which are very new and really not validated in humans, plenty of companies doing those. Moderna were the joint first to vaccinate a human being, jointly with CanSino in mid-March. BioNTech, a very dynamic and enterprising biotech company. CureVac have been doing RNA for many years. They're not quite in the clinic yet. Robin Shattuck at Imperial College London. These are all RNA vaccines. They're a bit more different than the viral vectors, but again, if one works, maybe many of them will. They have some manufacturing challenges, not in speed of manufacture. They were first into the clinic with RNA, Moderna, but in terms of scaling up and just getting the nucleotides they need to manufacture RNA vaccines. And then there's Inovio with a DNA vaccine, which is related, of course, but they need an electroporation device to get good immune responses. Then going back in time, if you like, the classical vaccines, of course, are protein in adjuvant, with GSK and Sanofi, the world's largest vaccine manufacturers, coming together to collaborate, but they take longer to make. Look at the timeline for that vaccine, mid-2021. And then there's a biotech in America, Novavax, who are really moving, and they should be in the clinic by July. And then, of course, a vaccine type that would have been familiar to Louis Pasteur, since he invented it for rabies, is an inactivated whole virus vaccine, where you do need to grow the virus and kill it, inactivate it, and then mix it with an alum. Those are really moving as well. 2 Chinese companies, Sinovac with good data in macaques. They're the only people I know who have macaque efficacy, as well as the FADOX1, and Sinopharm, they've been in the clinic for the last month as well. So it's a diverse portfolio. It's fantastic for vaccines that these technologies are really being stretched to the limit to see how quickly they can be manufactured and at great scale. And no matter what happens, we'll learn a lot about the relative merits of these different vaccines as the months go by. So just summarizing the current plan for our vaccine, we've had the first batch made at our own in-house facility, and we've got some more material from there. The group in Rome used a slightly different wave bag process, but making moderate-sized batches, going up to 200-liter scale. Now, 200-liter is 200,000 doses. If you get 1 per mil, if you get 5 per mil, that's 1 million doses. If you get 8 per mil, it's more. But look at what you can do in China or in India. 2,000-liter bioreactor scale is being developed in collaboration with us at WuXi, at the Serum Institute of India. These are all contract manufacturing organizations scaling up at risk, so that if the vaccine works, they'll be ready to press the go button and manufacture at great scale at 10 million liter bag or batch amounts. And then most excitingly, of course, in the last 2 weeks, this was only finally formally announced at the weekend, the university has agreed a contract with AstraZeneca, where we have out-licensed the vaccine for them to develop at great scale and globally, and that seems to be going extremely well. For example, just today, they've announced a BARDA contract of about $1 billion to make 400 million doses of this vaccine as fast as possible for the US. Already the UK government had booked in for over 40 million. The number keeps increasing. It probably will be more doses of vaccine for the UK. And of course, we as academics have been extremely concerned that the rest of the world doesn't get left out, particularly low-income countries. And indeed, there are contractual requirements for AstraZeneca to do this, and they will do this with CEPI and make very large numbers for much, if not all, of the rest of the world. So on a cartoon, if you like, this is what Sandy Douglas put in place back in early April. Feels like a very long time ago now, working with Paul to develop and optimize the process, transferring to Helix and Cobra, those contract manufacturing organizations, later on Oxford Biomedica, and then the big boys, Serum Institute of India and WuXi, and others now in the United States working with AstraZeneca, who of course have a base there as well as in Cambridge, England. So in terms of scaling up, it has gone extraordinarily well and quickly. We are not there yet. No commercial vial has been filled yet, but the target of September is looking good. Why is it September? Because we don't realistically think we could have any approval even for emergency use before September. You think about the efficacy result, we're hoping to have that by August, but there is no guarantee. So September is a reasonable target date for tens of millions of doses for the UK and many more for other countries and other parts of the world. So what could possibly go wrong? Well, about a million things, but let me highlight some of the challenges. Firstly, the most severe COVID disease is seen in older adults. Most of those who die are over 70 or even over 80 in some series. So vaccines, sadly, don't perform typically as well in people over 70 as in 25-year-olds. Immunogenicity can drop or may not, we just don't know, and that's being tested very, very soon. But that may be an issue. We may need a second dose in adults, we may need an even higher dose, or it may not work at all. We just need to wait and see. Of course, we don't know if the vaccine will work in younger people, but as Sarah Gilbert announced, she's about 80% confident that it will. And she's probably the best judge of anyone. How long will the protection last? Well, we don't know. The good news is we don't need it to last for 5 or 10 years. Lasting for a year would be pretty useful. And we will, of course, follow that. We are asked, will you need to have this vaccine seasonally like a flu vaccine? Possibly, but not in the same way that you need a flu vaccine. So what I mean by that is if you look at the genetic diversity, and lots of people have, of huge numbers of different coronavirus 19 sequences, the spike protein, yes, it does vary, but the variation within that gene is minimal compared to the variation you see in, say, HIV or HCV or even in influenza. So we're nowhere close to needing 2 different strains of COVID vaccine, at least right now in May 2020. Of course, in 5 years' time, after there's been a lot more diversification, that might be an issue, but certainly for the coming winter season in Northern Hemisphere, 1 strain will do. Then there's been some discussion about whether we need a vaccine to prevent people dying or getting unwell, an anti-disease vaccine, one to prevent transmission from one person to the other, or both. And of course, the answer is obvious. You would like a vaccine that does both. It's often harder to make a vaccine that prevents transmission than disease. I think we'd be happy with an anti-disease vaccine that kept people alive, at least in its first iteration, at the first try. But we actually think this will probably be a pretty good transmission blocking vaccine as well. But we'll be able to find that out before too long as well. If there are some of those problems, what can you do? You can try and redesign your antigen in the vaccine with a better version of the spike protein. Many people are choosing a fragment of it called the receptor binding domain, which is clearly functional. It gets the virus into a cell. You can try and restructure that and stabilize it, as was done with the respiratory syncytial virus vaccine. That doesn't seem to help, so far at least, with COVID. Obviously, you could try extra doses. That often doesn't work so well with a vectored vaccine where you get some anti-vector immunity, but often you do see boosting at, say, 4 weeks, or you certainly do at 3 or 6 months, so that may be helpful. If you do a different strain of adenoviral vector, say Ad26 rather than Chad, that will probably boost better than a homologous, at least at a short time interval. And then, more interestingly, you can deliver these viral vectors by aerosol. That's been done for example, tuberculosis by Helen McShane's group at the Jenner Institute, and works really well. It's safe and it gives very strong intrapulmonary T-cell responses and mucosal immunity. And that might be what you really need to prevent this lung disease. And we certainly plan to look at that with the Chad vaccine. It hasn't been top priority because it's a bit more time consuming and complex to deliver an aerosolized vaccine, even with an asthma inhaler, but that will happen reasonably soon. And then, of course, what the media have really picked up on is this idea of doing a challenge study, where to stop worrying about whether there'll be enough cases, you make your own and invite young, healthy people to have an inhalation of a certain dose, we don't know what dose, of SARS-CoV-2 after you have vaccinated them or not vaccinated them, and see in that way if your vaccine works. Now, that consensus view seems to be that that may well be okay, as long as the people are very healthy, pretty young, you get the right dose of virus to give them, and probably most importantly and least available, you have some good antivirals to treat them. Now, a month ago, we certainly didn't. In the last month, we've had remdesivir, we've had interferon, a couple of other antivirals that probably in combination are going to work really well, particularly against a low challenge dose of virus. So personally, I think this challenge model will come. It's not quite there yet, but in a year's time, it might be the standard way of comparing different candidate vaccines and looking at the durability of protection and mucosal immunity and all sorts of other things. So I think that should and will be valuable. So we're just about out of time. This has been an extraordinary experience for all of us involved. It's been fast moving, to say the least. It's a very different way of developing vaccines. I would suggest it's a better way of developing vaccines. And of course, we're always asked, why don't we make vaccines this fast for other diseases all the time? And the short answer is we'd love to, but what we can't do is persuade people to start sending us money, which they literally have been doing in a philanthropic sense in the last few months. We can't get agencies to prioritize one disease normally. They have in the last few months. We certainly can't get regulators to put our application at the top of the pile, ethical committees to meet specially, et cetera, and take out all the delays that you normally come up against in developing a vaccine sequentially from mouse to monkey to humans to phase 1, long delay, apply for money, phase 2, very long delay, go into phase 3. The point is you can do it faster than we are doing it if everybody pulls together. And I would submit there are some diseases where the world should do that, maybe for HIV vaccine or for a malaria vaccine, which is my favorite, which has been delayed too long. I mentioned that it's not just a UK activity, there will be trials outside the country. We can't confirm where they will be yet, but they are in late stage discussion and efficacy there may be key. Global supply is a huge challenge. People throw around huge numbers, not realizing that billions have never been done before, and not everyone's aligned on who should get the vaccine first. I haven't even touched on that. If you've only got 50 million doses, where do they go? I don't think we'll have that problem. We're lucky with our manufacturing scale, but some other groups certainly could. And of course, there are lots of questions, as you know, about the future course of the pandemic, some of it controllable by human action, some of it probably not. And that may well impact on whether we end up soon with a vaccine, or indeed never with a vaccine, or something in between. So I'll close by acknowledging again some of the senior people in the Institute, Sarah, Teresa Lamb, Katie Ure, I haven't mentioned, leading the immunomonitoring, in which by thousands of people, quite a challenge. Sandy Douglas on manufacturing and Pollard, the chief investigator of both the clinical trials I've mentioned. But most importantly of all, there are literally hundreds of people who normally work on other vaccines in the Institute who have swapped over to help with the extraordinary COVID-19 vaccine response in the Jenner Institute, in the trials group, the Oxford Vaccine Group, and at the Clinical Biomanufacturing Facility. The out-licensing to AstraZeneca was run by Oxford University Innovation, our tech transfer company. We needed the manufacturing partners who started at risk to get us where we are today. We have Vaccitech, who started out being partners in this, and AstraZeneca, who have ended up being our major industrial partner and have been fantastic so far. And crucially, we've had a large amount of support from the UK government and various agencies of it, and the Coalition for Epidemic Preparedness Innovation. So I will stop there and thank you for your attention and take any questions.
Thank you, Professor Adrian Hill, for that wonderful talk. And now